folks. Let's begin the lecture, guys. If you could quiet down and get your seats. I'd like to start the topic so that we have enough time to cover it during the 50 minutes we have. Last time I talked to you and introduced you to the angiosperms, and I said they are, in our minds, the most recently evolved group of plants, and that they became very successful. They essentially have colonized all of the earth and have really displaced in many areas most of the previous plants which had evolved before them. And we said that the two main reasons for the success of the angiosperms, among the many, was that they produce flowers, and the flowers help with the transfer of pollen, and we talked a little bit about that last time. And that secondly, that they produced a structure which is known as a fruit, which we said in most cases was an enlarged, although not always, ovary wall. Today what I'd like to do is to continue talking about the angiosperms and to take the beginning of the lecture where we ended last time, that is with the double fertilization, that process which gave rise to two types of fertilizations. And just to take you back to the female gametophyte, this should all be a review for you. And to focus on the two cells, or the two groups of cells that are important for our discussion, we said there were two N nuclei in the center, which we call the polar nuclei. And there was the egg. And now once we get this fertilization, we get the beginning of the seed that is starting to mature. The egg develops into the 2N embryo. And the really remarkable event which happens is that when the new nucleus from the double fertilization comes in and joins the polar nuclei, all of these nuclei combine to make a 3N tissue which surrounds the embryo, and that tissue is called endosperm. So this then is the status of a seed as it's maturing. It has the seed coat on the outside, and then within the seed we have the developing embryo, that is the new sporophyte, and this tissue, this triploid tissue, which we're going to talk a lot about, and with which all of you daily have contact. You may not realize it, but you do. Now, what I want to do with today's lecture is to begin a discussion with you on the biochemical and physiological controls of plant development. Very fascinating subject. There's a lot of controversy still, a lot of things we don't know, a lot of contradictory things, as you'll see as we go through the subject matter. And one of the most important sort of foundational understandings that you have to have when we talk about plant development is that once the seed lands someplace in the soil and puts down its roots and starts to grow, that plant which comes from the seed will spend its entire life in that spot. If the weather turns bad, or there's a lack of nutrients or water, or another tree grows over and shades it from the sunlight, this plant can't get up and move like an animal can. It is essentially fixed in this one position. And as a consequence, plants have developed mechanisms, extremely sensitive mechanisms, for measuring their environments, much more sensitive than we are. They can measure, they are aware of things in their environments that we can't even guess. Secondly, the plants have developed very unique mechanisms for adapting to the changes in their environment, since they can't get up and move. They have to adapt some way or die. And we're going to talk a little bit about the signaling mechanisms, the perception mechanisms that are involved to sense the changes in the environment. And also we're going to talk about some of the responses. But as you might guess, this is a topic which is extremely interesting, and also there's a lot of information on it. Now, to begin our discussion of plant development, I want to say to you that one of the main things that we have to keep in mind is that when you study plant development, even at a level that we're going to talk about here in class, which is kind of a superficial level, you've got to know something about the anatomy, the cell types which make up the inside of the plant, and also something about the structure, the outside structure of the plant. So I'll be giving you a few new terms. I'll try to keep them to a minimal, but we're going to need them in order to really appreciate this remarkable system of development that the plants show. And I'd like to begin by talking about the angiosperms. And the angiosperms will occupy now the lectures for the entire period of time on plants. And when we begin our discussion of the angiosperms, which we said numbered about 240,000 species, a large group of plants, when we begin this discussion, in order for us to simplify it somewhat, I want to make a division of the angiosperms into two groups. And this division is based on characteristics of the embryo. That is, the embryo inside of the seed. So let's start by saying that there are two types of embryos. And I'll draw the first type, and then we'll label the parts, the structural parts that are important for our understanding. The first type of embryo, which we see in the seed, and don't worry about the preciseness of the drawing, there are a couple of things I want to indicate, has on it two bumps in the embryo, two bumps. These bumps form what are known as the seed leaves. They are present inside the seed. They are the seed leaves. This would be the embryo axis. And we give a name to these seed leaves. They are called cotyledons. Cotyledons. And because there are, in this particular categorization or division, two bumps, we say that this seed represents a plant which is a dicotyledon, or a cotyledonous plant. So this is frequently shortened, as I will in lecture, to dicots. That is, embryos from the angiosperms which have these two seed leaves or two bumps present inside the seed. The more modern term for dicots, which your textbook uses, is eudicots, just EU added onto it. So one group of the flowering plants of the angiosperms is called the dicots, and it is based on the fact that there are two seed leaves present in the embryo. The second division also has an embryo axis, just like we saw before. So here's the axis. The embryo axis. But it has only one bump. And it's often on the side, as we'll see in a moment. And these are known as monocots, or monocotyledonous plants, because they have one cotyledon, a monocot. Now let's look a little more closely at the embryo axis just before the seed germinates, and we may have to kind of stretch it out a bit. And we're going to draw now the embryo axis, and I'm going to draw a eudicot embryo axis. So here is the axis, and at this point we're going to put on the two cotyledons. Cotyledons on the axis, and a couple of landmarks that you need to know. The region above the cotyledons, because we're going to talk about this, is known as the epicotyle. Above the cotyledons. And the region below the cotyledons until we get to the root is known as the hypocotyle. Just to be able to talk about whether you're above or below the cotyledons. And this is important for a lot of developmental work. Now, the other two landmarks I want to put on this embryo axis, which are probably the most important point for today's lecture, are the following. And I'll write this down for you. At either end of the axis are regions of high cell division activity. High cell division activity. And we call these regions meristems. And there's one located at the shoot end. This is known as the shoot apical because it's at the tip, meristem. And there's one located at the tip where the root would be, which is the root apical meristem. So these are regions which have a lot of cell division activity going on. And the reason I am making an important point about these meristems is the following sentence. And this is probably the key top point of today's lecture. If, for development especially, if a newly formed cell can trace its origin to division activities of either meristem, that new cell or cells are said to be primary. This is important because, as you might guess, if we have the word primary, that means there are going to be other ways of originating cells. So that if you can find a cell over here, say some distance down from the meristem, which arose because of division activities in the shoot apical meristem, that cell or tissue is said to be primary. An important point. Now what I'd like to do is to talk to you a little bit more about seed physiology. In the diagram that we have above, we showed that the two products of double fertilization were the new sporophyte embryo and this triploid tissue called endosperm. What we can say at the outset is that endosperm functions in providing nutrition to the developing embryo. So here we have this seed, which has the small embryo, and also that embryo is surrounded by this triploid tissue known as endosperm. That embryo can follow two pathways. In one pathway, the endosperm is absorbed into the cotyledons. And so if you were to look at a seed, and we'll show you one in a moment, of the type where the endosperm is absorbed, what you would find is that the seed is essentially filled with a very small embryo axis, but mostly cotyledons. The endosperm has all been absorbed. And this is typically the situation, although not always, in eudicots. If you were to open the seed of a eudicot, most of the endosperm, which had resulted from double fertilization, would have been absorbed into the cotyledons. And this is contrasted with monocots, in which generally the cotyledon is small and endosperm is still present. Now, I said when I introduced the term endosperm to you that it's something that you have contact with every day. It's something with which you are familiar with every day. And we can expand that statement by saying that both endosperm and cotyledons function to nurture the establishment of the seedling. Probably the most critical time in the life cycle of a plant is when that seed germinates and has to produce a root, has to get from the soil where it's buried deep without light and expose itself to sunlight so it can begin to undergo photosynthesis. Until it does begin to undergo photosynthesis, its nutrition, the material that sustains it, comes from either the endosperm directly or from endosperm that has been absorbed into cotyledons. You daily eat endosperm, eat the cotyledons, because, as you will see from our demo in a moment, the endosperm and the cotyledons are the main portions of seeds that you consume. Beans, peas, all of the legumes and all of the plants that you eat are a combination to get from the seed, a combination of nutrition from endosperm or from cotyledons. So this is where our major supply of food comes from. Now what I'd like to do is to give you some examples of some of these seeds, and I'd like to begin with corn, which many of you are familiar with. Most of you who are gardeners or who have a garden at home know that if you harvest the corn and immediately cook it, or if you buy corn, for example, from Berkeley Bowl or Monterey Market, where it's often refrigerated, that when you bite into the corn seed, and in fact, as you'll find out later, these are really fruits, but for our purposes here, let's call these seeds. When you bite into the corn seed, you will get sprayed on your face by this white milky liquid. This white milky liquid is endosperm. This endosperm functions to sustain the developing embryo in the corn seed. Now, I want to present you with another question, and I'll set this question up with the following situation. Normally in nature, the liquid endosperm, because it is liquid, when you see it squirt out, the liquid endosperm is high in sugar. Now, in nature, if this ear should fall to the ground, or for whatever reason you have not picked it, what we find happening as winter approaches is that endosperm is converted to starch. And this frequently happens if the corn has been sitting around, for example, in Safeway for a long time. You go to eat it, and it's not sweet anymore. That's because the endosperm has been converted, the sugar in the endosperm converted to starch. So let's go back to this ear which is laying on the ground in the wintertime. The stored nutrients are now in the form of starch, and then what happens is, as spring comes along, the starch is converted back to sugar again, back to a liquid again. You could taste it. It probably wouldn't be very good, but you could taste the sweetness. And my question to you is, why go through what are really quite complicated and energy-requiring biochemical processes to convert your stored nutrients from sugar to starch and then back to sugar again? Why does the plant do this? What is the advantage, or what is the reason for this conversion? Now, another example of endosperm that probably many of you have had experience with is the coconut. If you take a coconut, I don't think you can hear it, but the liquid inside the coconut is essentially liquid endosperm. So if we take a coconut, and this is a large seed, and on the outside there's part of the fruit wall. So the seed coat and the inner part of the fruit, or the ovary, are what make what you think of as the coconut. This is what the coconut would look like if you were to get it directly from the tree, where the fruit wall would be considerably larger. Let's take this coconut now and make a cut down through its center and show you the various parts of this particular seed. So, the coconut is a monocot. And one of the ways you can tell that it's a monocot, and you will see this in the lab, and there's no necessary reason for you to write it down, is that the monocots, their flowers and their fruits are in parts of 3. And if you look carefully at a coconut, and I encourage you to do so next time you go to the grocery store, or you'll see from the pictures I'm going to show you, you will see that there are 3 depressions, or people call them eyes, at the one end of the coconut. And if you look carefully, you will see sutures separating the eyes from each other. These 3 eyes with the sutures represent 3 carpels, which have fused together in development. So the coconut fruit or coconut seed consists of 3 carpels that have fused together. Now let's cut this coconut down through the middle. Cut it down through the middle. And the first thing we see is the liquid endosperm. Liquid endosperm. These are 3N nuclei just floating around in the liquid. For those of you who come from Asian cultures, or from those of you who are health fans, liquid endosperm can be bought. It is an extremely rich mixture of, so far, uncompletely identified nutrients. A lot of amino acids, a lot of proteins, which serve to nourish the developing embryo. The embryo would be located at this end. And in this particular case, the single cotyledon would be small. This is a monocot. Most of the nutrients for this seed are stored in the endosperm. Now, most of you, when you buy coconuts and you open them up, you find that there's a white material, which is really what you want to eat, all the way around the periphery or perimeter of the coconut. That white material is endosperm, which has solidified. Each of the nuclei have been partitioned off into a cell. So, in this particular case, if we take this coconut and we cut it open, what you can find, well, that's only part of it, what you can find is we have the coconut that you guys eat. The seed coat is here on the outside, this brown material. This is the fruit wall, and part of the fruit wall is also on the outside of the coconut. The white material, which we have here, is solidified liquid endosperm, and the embryo, which we're going to look for now, is also in this structure. So let me just get these guys before we get any more endosperm here. And what I'm going to do, for those of you who are in the front, I'm not sure you want to sit there. Going to open one of the coconuts up. No, you don't have to leave. I'm just kidding. Well, I might or not. So, all right. So I just happen to have a coconut that's open. So you don't have to worry about it. This is like the people who do the cooking, they already have everything cooked for you. And we've opened up this coconut, and when the coconut is opened up, I've already drained out the liquid endosperm, but you can see the solidified white endosperm, which is surrounding the coconut. Now, this has an embryo inside of it, but the embryo is not present where you might guess it would be. Because this is a monocot, and because it had 3 carpels, each of which had 1 ovule, all 3 of the ovules were fertilized. So we had 3 embryos developing in the tiny coconut fruit. And then what happened is the plant said, I have 3 embryos, they're all good, but I only have enough nutrition to sustain 1 of them. So I'm going to abort 2 of the embryos. So if you look under where the eyes are, you will see 1 quadrant is larger than the other 2. Under the eye of that quadrant is the embryo. And under the eyes of the other 2 quadrants, there are aborted embryos, which you cannot see. And here up front, I've taken out the embryo from the coconut that I've already opened here. It is a structure which is about a half a centimeter big. I'll show you a picture of it in a moment. And again, these are boring party tips. If you're ever at a boring party and they have coconuts, you can become the center of attraction by finding the embryo, okay? So, this is a coconut, a very, very important food crop, and it is a crop that exists because of the endosperm which exists and which we eat. Now what I'd like to do is to have everybody take the peanut that we gave you. And again, these peanuts haven't been roasted, so I'd prefer that you not eat them. In fact, it's probably safer. If you're allergic to peanuts, don't open it up. That's okay. The very first thing I want you to notice before you open the peanut, and try and save the shells or else I get in trouble with the custodians who come only once a month. What I'd like you to do is to look at the outside of the peanut. The outside of the peanut is surrounding the seeds. The peanut is an angiosperm. That outside structure now is protecting the seeds on the inside, the things that you eat. And that outside structure, because it is protecting the seeds, and if we trace back where it came from, we would find that at that outside shell, that you call it, is ovary. So in this particular case, the ovary wall, which in the apple was soft and sweet, has become hard. So here we have a hard ovary wall, which is now surrounding, and I'd like you to open it up, surrounding the peanuts inside. So if you open it up, you're going to have a peanut on the inside. And you'll notice that the peanut on the inside has a little brown, papery cover over the individual peanuts. This is the seed coat. And now what I'd like you to do is carefully split the peanut in half. So you have the two halves of the peanut split in half. Each of these halves is a cotyledon. So this is a eudicot. And if you have split the peanut in half carefully, if you look at one end, you will see a little difference at that one end. And that difference is the embryo, or the embryo axis, located at one end of the peanut. So these cotyledons are what we eat, will provide us our nutrition. When you eat something like a walnut, again, the hard outer coating of the walnut is part of the ovary wall. And so if we open the walnut up, we looked on the inside of the walnut, the things that you eat for the walnut are cotyledons. This provides the nutrition for humans, but more importantly, it provides the nutrition for the developing embryo until the plant is able to photosynthesize. So this is an important characteristic of the plants as far as their ability to store minerals and nutrients inside of the seed, and thereby are able to establish themselves and get going by allowing the plants to send their roots down and then for photosynthesis to occur. Let me now show you a couple of pictures of the coconut. Oops. The first slide is meant to show you the coconut typically to what we have up here. And what I want you to notice on here is that we have three eyes. This largest eye is the one under which you would find the embryo. And if we look at the embryo, again, it would be under this large eye, this large quadrant, which represents one of the three carpels. Here's the embryo of the coconut, and I have one up here in front that you can look at after lecture. It is about half a centimeter to a centimeter long. One end of this embryo grows into the liquid endosperm and begins to absorb the materials for the established coconut. Here we now have a coconut which is producing the endosperm that has solidified and become hard, and this endosperm now is filling the coconut, and the coconut shoot system is using the endosperm until it is able to get established. Now, what I want to do is just blank the slide for a moment. We'll leave the screen down, and what I want to do is to say to you that although we said that the great advantages to the angiosperm were the development of the fruit and the flower, and now we have this mineral nutrition that's stored in the seed, there is also another advantage that the angiosperms develop, which allowed them to be so prominent and so widely spread. And that advantage is that the angiosperms have developed mechanisms to disperse their seeds and fruits. And I'd like to briefly just mention some of these mechanisms that the angiosperms have, and then we will look at some pictures to demonstrate them to you. These mechanisms ensure dispersal of the seeds over large regions and make sure that they are carried off into new areas to allow the plants to colonize them. The first mechanism depends on wind. Many seeds have like dandelions, have like almost like a parachute on them. They're small and light, and the wind distributes them. Many seeds are dispersed by water. And up front here I have a good example of a seed which is dispersed by water. Here is the coconut, in which the whole seed and the fruit float. And they can float considerable distances from one island to another, containing the nutritions in the endosperm. The outside fruit wall is able to resist the salt water and thereby protect the developing embryo. So water is often a great way in which the seed is dispersed. Another mechanism is mechanical. Many seeds are found in fruits that if you touch the fruit, all of a sudden the seeds shoot out. They are propelled away. And this is probably used as a way of dispersal when an animal walks past and rubs against the fruit. It disturbs it and causes the fruit to disperse and shoots the seeds off into the coat of the animal, who then distributes it someplace else. Another mechanism for the dispersal of seeds has to do with animal, but mostly insects. Many seeds, in addition to producing an ovary wall, making a fruit, also make another structure which attaches to the seed. We don't have to worry about the name. We just say it another structure which attaches to the seed. And this other structure is particularly attractive to insects who harvest the seeds, and this is so with ants, and I'll show you a picture of it in a moment. Harvest the seeds, take the whole seed with this accessory structure on it back to their burrow, consume the accessory structure, and then the seed is already buried in the ground and distributed to some other place. And finally, a mechanism of dispersal of the seeds that many of you are aware of already has to do with the seeds that stick to your pants or to your socks when you walk through a field or when you walk through an area which is highly grown with a lot of plants. This sticking to your clothing is because the seed contains barbs on the outside, and I'll show you a picture of it, or the fruit contains barbs on the outside. And for those of you who are interested in a relatively recent discovery of how seeds can help us aside from being sources of nutrients, these barbs, in somebody's mind, he looked at them, and this is the way Velcro was discovered. The person saw that you could continually take these on and off by having the barbs release and then join back on to some other surface. So this is an example of a field which is known as nature-inspired design or biomimicry. A very hot field these days. What can nature tell us about our design? So now let's take a look at some pictures of seed dispersal mechanisms. And the next slide will show us a picture of a plant that's growing here on campus called impatiens. And this plant over here produces a fruit. When the animal comes by, the fruit is usually swollen, and the animal touches the fruit, and within milliseconds, the fruit discharges, and the seeds are propelled in up to 10 meters. This is also found in another California native called salvia, where when you touch the sepals from the outside or the calyx tube, the seed shoots off sometimes up to several meters away. Here's an example of wind dispersal. This is milkweed, in which this umbrella or parachute carries the seed sometimes great distances. Here's an example of wind dispersal, but a wind dispersal of the entire plant. Those of you who come from the Southwest probably have seen tumbleweeds before. In this particular case, the tumbleweed is severed from where it joins the root, and then the wind pushes the whole plant, which rolls, and as the plant rolls, it distributes the seeds over a large area. Here is an animal, an ant, eating that accessory. Here is the seed over here, and here's the ant selecting the seed and harvesting it and bringing it back to its burrow in order to consume this accessory nutrient-rich structure. Here's a seed with barbs on it, the basis for Velcro, or actually is a fruit there. Here is a seed that many of you know. This produces an accessory structure, this seed, which makes the seed particularly attractive, probably to a large animal. And this is the origin of nutmeg and mace. The red material which covers the seed is an accessory structure. Here's the ovary on the outside. and the seed or the nutmeg is on the inside, and this red structure, which is also nutritious and attractive to some animals, is consumed and allows for the distribution of the seed by what must be a fairly large animal. Here is a seed now which is meant to be attractive to birds, in which the outer portion of the seed has developed this red structure to be attractive to the birds, maybe also sweet. Here we have a seed or a fruit where there's a contrast between the seeds and the fruit to attract the attention of something who would distribute it. For those of you who come from Florida or from a tropical area, there are these ultramarine seeds, the very first seeds I've seen that are sort of blue-green or turquoise in color. And I don't know what animal would be attracted to this particular color. And finally, a seed which is distributed by animals and also by ants and other insects. The red is the ovary wall, the black is the seed, and this white structure around the outside is this sort of accessory tissue, which is used for the development of a mechanism to be dispersed. Okay. Now what I'd like to do, yes? How do you define an accessory structure? Accessory structure is something which is formed between the ovary wall and the seed, and it represents a new growth. It's given a technical name, which I'll give you, but it's called an aril, A-R-I-L, but it's not necessary that you include that in your notes. Now, once the seed germinates, it's going to start to be established. So the seed has been distributed, and now it's got to germinate and establish itself. And let's look at the problems a seed is going to have when it begins to germinate, when it begins to start to produce the shoot system. So here we have the soil surface. Here we have the soil surface. And at the soil surface, we have light, and we have fresh air, and maybe the seed finds itself located somewhere deep in the soil. This seed has got to make it up to the top of the soil in order to begin to photosynthesize. Now you can imagine, if this seed is going to germinate in the soil, that it may not be a clear pathway up to the top. There could be, for example, a rock here, or the soil could be very hard. It could be clay-like. So let's go back to our seedling axis and spend a couple of minutes just looking at this axis and talking about the problems this axis is going to have in order for it to break the soil surface. Here we have the cotyledons, which we mentioned before. And let's look at a dicot, the cotyledons. And if we focus on the two regions that are very sensitive, we look at the shoot meristem in this particular case. Now, the problem is if the seedling was going to germinate and grow up to the top of the soil with the shoot end growing up, this meristem would be impacting on all of the obstacles in the soil, including rocks. And the meristem could be easily damaged. So what seeds do, and this is why I introduced you to the terms hypocotyl and epicotyl, is the following. When they germinate, they're bent over into a hook formation. And I want you guys to notice this. Here are the cotyledons. Here is the shoot apical meristem. And what is putting pressure on the soil is the hypocotyl. It is acting as a battering ram, just like your elbow pushing up through the soil with the meristem being your hand. So the seedling then pushes up to the top of the soil. Once it sees light and breaks the surface of the soil, this hypocotyl region has served its role of acting as the battering ram, and the seedling then straightens up, and the shoot meristem no longer has to worry about running into any obstacles. This is what you would see in a eudicot. How about a monocot? Monocot's a little bit different. And let's talk about seed germination in the monocots. Seed germination in the monocots. Here we have the seed. Here we have the soil surface. And the monocots do it a little differently. If we look at the monocot axis, if you look at the monocot axis, so this is the axis of the monocot, it grows straight up. It's not bent like we see in the eudicots. But the monocot covers its shoot apical meristem, which is here. Covers its shoot apical meristem with a, if you want to call it a hood or a sheath, which becomes the battering ram, and the shoot apical meristem doesn't then contact the soil directly. The reason I want to introduce you to this hood or this sheath is that we're going to see it later on, and the technical name for this hood or the sheath is called the coleoptile. And we'll not say any more about it here, but I want you to know what part of the plant it is and where it comes from. Now finally, what I want to do, as I mentioned at the beginning, is to put a few landmarks on the plant that we're going to be referring to, terms that we're going to have, not many, but you need to know what they are and to put them into your notes. Here is the axis again, the axis of the plant. And let's put some of these landmarks on now. Here we have the leaf. And the leaf is divided typically into two parts, into the blade and into the petiole. And there might be another leaf on the other side too, divided into the blade and the petiole. And these can be variously modified depending upon the plant. At either end, we have the meristems. This is the root meristem, which I'll just call root apical meristem. And then we have a few other things that I want to put on here. The region of attachment of the leaves is called the node. It's called the node. So any place a leaf is attached is a node. And if we have several leaves on here, let's just say we have several leaves, region between leaves we call the internode. Now what I'd like to do is just make a little high power view of one of these nodes, because there's one other item on this shoot system, this vegetative shoot system, that you have to know. So here is a high power view of a node. Here's the node. If you look carefully at a node, what you will find in the region between the main axis and the petiole, which is called an axil, you could call it lots of other things, what you will find in this region is a little structure. Very conspicuous sometimes, sometimes very inconspicuous. This is a bud. This is a bud. And the bud becomes the branches. So each of these branches, which is growing out from this particular stem here, is growing out at a node where at one time there would have been a leaf attached. And each of the buds potentially can grow into a new branch or a new axis. We'll talk more about this when we talk about development. Because this bud is in the axil of a leaf, we call this bud an axillary bud. An axillary bud. Finally, on this particular plant, this is vegetative. It is not producing any flowers. But in the shoot system, one could also have flowers, and ultimately one could have fruits. So depending upon the age of the plant, one could also have it in a reproductive state. Let's look now at a few pictures of these particular events. First slide is going to show you the plant that we've just talked about, where we have the apical meristems at either end of the axis. We have the leaf with the petiole, and in the axil of the leaf, we have the axillary bud between the two nodes, which we said were called the internodes. These are probably all you have to know of with regard to the shoot structure. Now here's a germination that I talked to you about of the eudicots, which is germinating beneath the soil. And notice this hook, this hypocotyl hook. The apical meristem would be down here, protected from the battering of the soil. And once it breaks the surface of the soil, the shoot system then straightens up, as you can see in this slide of a real plant, in which the hypocotyl hook is bent, and once it sees light, it then straightens up. Here is the monocot now, and here in this particular monocot, we have the structure which is the sheath that covers over the tip of the shoot apical meristem, and this sheath thereby provides protection to the developing shoot apical meristem. So these are mechanisms which are used in the establishment of the seedlings. Now, before I end the lecture today, I wanted to remind you, a note I put up on the board before. Next week, you are asked to collect both a weedy flowering plant and what you think of as a gymnosperm cone. Those are necessary for you to bring to the lab in order for you to complete the exercise. And secondly, your GSI asks that before you come to lab next week, you look at two things which are on B courses, I guess they're called. And they are two videos of the reproductive life cycle of the ferns and of the angiosperms. It'll really help you with the lab. Have a nice weekend.